Welcome back, everyone, and congratulations, you made it past the plants. I know based on your pre-course survey responses that many of you were not enthused about going through plants, but you made it through. And now for the rest of the semester, we're going to be talking about animals. So we'll begin with animal diversification and some information on how they interact, and then we'll spend the rest of the semester talking about animal anatomy. So let's begin this third section of the semester with Chapter 13, discussing animal diversification. So before we really get into animals and the different types out there, we should first define what an animal is. So animals are multicellular eukaryotes and they're heterotrophs. This means that they must consume other living organisms in order to get their carbon. That can be other animals and that can be plants. Animals are also motile. That means that they move at some point in their life cycle. Many of them move as adults and as young. Some are sessile as adults, meaning that they attach to one spot and they don't leave that location once they're mature and they're adults, but their larvae will be motile, and we'll see that in sponges in a couple of slides. Animals also all have a blastula stage in their early development. And a blastula is basically a hollow ball of cells in the early stage of their development. So after fertilization, the fertilized egg will begin to undergo mitosis, right, to form a ball of cells. And that ball of cells will have a hollow center, and then that whole thing, the ball of cells in the hollow center, is called a blastula. And that can be seen here in this image. So all of this purple material on the outside are cells, and then this kind of cavern in the center is that hollow middle. And all animals have this structure in their early development. Additionally, all animals undergo embryonic development. So they develop from an embryo into an adult. Animals also have additional features. They can reproduce sexually or asexually, depending on the species and sometimes depending on environmental conditions. And they also have specialized organs. These are organs that help with sensing their environment, avoiding predators, catching prey, defending themselves, or even feeding. All animals have a shared common ancestor, and this shared common ancestor was a protist, so an aquatic protist at some point hundreds of millions of years ago. And then some examples of some animals that you're likely familiar with are sea sponges, jellies, nematodes, insects, reptiles, amphibians, mammals. The list goes on and on and on. There are so many animals out there. And then there are animals that are extant that we all know of and we see all the time. And then there are animals that are extinct, which means that they have long since died off. And so you'll hear me say these terms throughout the presentation. So just remember, extant are animals that are currently living, and extinct are animals that have gone extinct. And all animals that are living currently today are equal to each other in the way of they're all successful. So no animal that exists today is better than any other animal that exists. And this is a common misconception that a lot of people have, is that humans are the most complex animal. Everything wants to get to be that. That is not true. Every animal is specially adapted for their environmental conditions. So every animal that exists today is successful. You will hear me use some terms rather than saying primitive or better or more advanced. We'll hear me say simple and complex, because organisms can have a more simple structure and more simple organs and systems, and then some are more complex. There are more parts involved. So when I say simple and complex, I don't mean one is better than the other. It's just one is more simply built and the other one is more complexly built. All animals on Earth are categorized using phylogenetics. And when you're using phylogenetic analysis, you really want to just understand the relationship between various organisms. Where do they share a common ancestor? Are their features come from a common ancestor, or are they analogous traits? So when we're doing phylogenetic analyses, there are a couple of different questions we can ask to group organisms into kind of larger groups. And so the first question we might ask is, do these animals have true tissues? And a tissue is just a group of connected cells that are all working together to perform a similar function. An example can be seen in this picture here. This is an image of human, I think, epithelial cells. And basically all those cells are interconnected and they work together to do a common function. And so most animals have true tissues, but there are some out there that do not. And one phyla of animals that do not have true tissues are the sponges. This is Phylum Periphera. And instead of having true tissues, they just have a group of cells, like an aggregate of cells that live together and they work together, kind of, sort of, but they're more independent. So just imagine instead of all these cells are the same type of cell working together to form a particular function. Instead, if you had a group of cells that all do their own independent thing, but somehow are able to work together to form an organism. So that's essentially the organization of sponges. Another question we can ask is, does the organism develop with radial or bilateral symmetry? And so what are those? Radial symmetry is when you can divide the organism with multiple bisecting lines and get two even equal mirrored parts. And so an example can be seen here with this jellyfish. So I can divide a jellyfish this way, bisecting it and cut it in half that way. And those two halves that I get will be equal mirrored parts. And then let's say I divide it in a different way. I divide the jellyfish this way. Those two parts that I get are equal mirrored parts. Same thing with bisecting with this line. I can get two equal mirrored parts. So it's almost like when you divide up a pie. You can cut a pie as long as you cut it in half. No matter where you cut, you're going to get two equal mirrored halves. And so some examples of organisms that exhibit radial symmetry are those that belong to the phylum Cnidaria. And this includes your jellies, your anemones, and your corals. Many of the organisms that exhibit this symmetry are slow-moving or free-floating, and they have no head and no anus. Organisms can also exhibit bilateral symmetry, and this is where you can only divide the organism using one line, so you can only bisect it one way to get two equally mirrored parts. And so you can see that with this frog here. I can only divide this frog from its head to its anus, so down this line here, and get two mirrored parts. If I would try to divide this frog this way, the two parts that I get, the two halves, would not be mirrored images of each other. And same thing with here or here. I can't just divide a frog in two halves anywhere and get mirrored parts. I can only divide it in half using this plane here and get two equal mirrored halves. And so this is bilateral symmetry. Bilateral symmetry is highly associated with cephalization, which is the development of a head. So bilaterally symmetrical organisms generally have a head. And most bilateral organisms that develop as embryos are bilateral as embryos and then also bilateral as adults. But there are some exceptions. So there are the echinoderms that start out as bilateral young, so their larvae are bilateral, but then they later develop into radially symmetrical adults. So this is one example of an organism that can switch between them, but they are bilateral organisms because they start as bilateral when they're larvae. Another important question to ask is, during the development of the animal, does the mouth or the anus form first? And to understand what this means, we'll first take a look at early development of animals. So all animals, as I mentioned, have a blastula stage of development, which is that hollow ball of cells. For all of the animals except for the sponges, that blastula will undergo an invagination event and form a pouch that looks something like this. That pouch will actually later become the gut. Where the pouch opens up to the environment, so this pore, this hole right here, if that hole, that pore later becomes the animal's mouth, then those animals are called protostomes. If that opening later becomes the animal's anus, then those animals are called deuterostomes. And so what generally happens in all of the animals except for the cnidarians, so except for the jellyfish, anemones, and corals, what will happen is that pouch will continue to extend to the opposite side creating a tube that runs the entire length of that ball of cells. If that pouch does not keep extending, then we will later call that organism, we'll say that they have a blind or incomplete gut. So that just means that there's only one way for food to go in and one way for waste to go out. It's the same opening. But the vast majority of animals actually have two openings, a mouth and an anus. If that second opening, which is located here, becomes the mouth, and that first opening was the anus, then those are deuterostomes. In protostomes, that second opening will become the anus because the first opening is the mouth. So, as I mentioned, protostomes develop mouth first, and the order is mouth, gut, anus. And some examples of protostomes include worms and mollusks. And then the deuterostomes develop anus first or mouth second, and they develop anus first, gut, then mouth. And some examples are mammals, reptiles, amphibians, and echinoderms, or the starfish and sea urchins. For the next question, we're just going to focus on protostomes. We're not going to be addressing deuterostomes in this question. So the question we could ask is, does the protostome grow via molting or through continued additions to the body plan? And so what I mean by this is, molting is when an organism will have a hardened outer coating, and they must shed that coating before they can grow bigger because that's a hard outer surface. That hardened outer shell helps them to be protected from all different types of predators and from various dangers and threats. However, it restricts the ability of the animal to grow. And so they have to shed that outer coating and then they can grow. And then they form a new hard outer coating. So an example can be seen here. This fly type creature is molting. So this piece here, this darker piece that looks like it's grabbing onto that leaf, is its old outer shell, its old cuticle that it has now shed, and then it will emerge as this soft, fleshy, vulnerable insect. And then slowly over time, it'll develop a new hard outer shell, and then if it wants to grow again, it has to shed that shell. So this is exemplified in lobsters, insects, and nematodes. And then you can have organisms that can grow by just adding new body segments onto them. And this is an example of like mollusks and annelids. So like when you have an annelid, which is like an earthworm, the earthworms, let's see if I'll try to draw one. An earthworm has, oh goodness, has many segments, right? And we'll see a picture of an earthworm later if you're like, I don't know what you're talking about. But it's basically a long worm that has multiple segments. If that worm wants to grow, they don't have to shed their outer skin. All they do is just add a new piece to their end. So they just add a new segment, and if they want to get longer again, they add another segment and another segment. And that's essentially how they just keep getting bigger and bigger. Some examples of this are clams, octopi, snails, slugs, oysters, and earthworms. There are many, many, many more important questions to ask when you're trying to group animals together, and then when you're also trying to establish their evolutionary relationship and find their common ancestors. However, the last question we're going to talk about in this class pertains to both protostomes and deuterostomes. And the question is, does the animal have a backbone? If the animal does have a backbone, we call them vertebrates. And this includes fish, reptiles, mammals, and amphibians. If the animal does not have a backbone, then they're called invertebrates. And invertebrates make up 95% of all animals on Earth. So the vast majority of animal species are invertebrates. And this includes sponges, jellies, insects, mollusks, and nematodes. But the term invertebrate does not represent a monophyletic group. So their shared common ancestor is not a recent shared common ancestor. You have to go very, very, very far back to find a shared common ancestor among all invertebrates. So the term invertebrates includes 8 lineages of different organisms, including 7 protostomes and 1 deuterostome. So there are that they share the commonality that they don't have a backbone, which is an analogous character, because it does not derive from a shared common ancestor, but they are very, very prevalent. And then all of the kind of phylogenetic analyses that are going on are always changing. As we get more technology and learn new things, these classifications change or become refined or get broken into smaller pieces or grouped together. So phylogenetic analyses, or these kind of groupings of organisms to see how they relate to one another, is always evolving and changing. So the first group we're going to talk about, getting into the specific groups of organisms, is the Phylum Periphera, and this includes their sea sponges. Like I mentioned before, sponges have no tissues, and organs are made of tissue, so if they have no tissues, then they have no organs. And instead, they're just a collection of cells. So there's a whole bunch of different cells that kind of basically work independently, and they cooperate together to make an organism, but they're all independent cells. And there are a lot of different types that make up a sponge, but two of them that you should know are the epithelial cells, and those make up the epidermis, or the outside of the organism, so the part that you can see. And then there are these collar cells, and collar cells are responsible for filter feeding, so the organism can get the food it needs to survive. Sponges, like I said, they're a group of interacting cells. So how do they maintain some sort of structure? Well, they have these fibers called spicules that basically act almost like a skeleton for the sponge and give it its shape and keep it kind of all held together in a way. And sponges exhibit asymmetrical symmetry. So we didn't really talk about asymmetrical symmetry in the last couple of slides, but basically asymmetrical organisms don't have any symmetry. So you can't really divide them in half in any way and get mirrored parts. So some of them have some sort of symmetry, but a vast majority of them are asymmetrical. And they're invertebrates, right? They have no backbone. They don't even have tissues. So as adults, sponges are sessile, so they stay in one place for their entire lives, but their larvae are motile. So when they, after fertilization, then their embryos develop into larvae, their larvae can be pushed by currents to other places, or they can swim a little bit. They're not strong swimmers, but they can move themselves through the water to different locations where they'll sit down basically on a rock or on debris or some sort of substrate, and then they'll stick there, and that's where they'll spend their entire lives. Sponges are filter feeders, so they basically get the nutrients they need from particles that are floating in the water. And how they do this is they have these collar cells, and the collar cells look something like this. They're a cell that has this kind of net at the top of the structure and this flagella that is located in the middle of the net. So that net type structure is this thing here, and then this is the flagella. And they basically take that flagella and whip them around really, really fast. And all these collar cells are kind of close to one another. So when they whip all their flagella together, it causes water to move into the sponge via the pores or the main opening at the very top. So the water will come in, and let's say it gets into a pore. Within this pore, you have all these collar cells. The collar cells, in addition to bringing the water in, are responsible for catching the food. So the food particles that are in the water will basically hit this collar, this netting on the outside, or fall in the inside. And then that's the bottom of the cell, this part of the cell, will then basically take up that food and bring it into the rest of the sponge. So just to kind of go through the steps again. So the collar cells have a flagella that's whipping around really, really fast, and they have a whole bunch of them that work together. And when they're all together, they're able to bring water into the sponge. The water also has small particles of food. Those food particles will get stuck to the netting on the collar cells and then fall down into the base of the cell. And from the base of the cell, the food is transported wherever it needs to go in the sponge. Sponges can undergo both asexual and sexual reproduction. The asexual form is through budding, so basically parts of the sponge will break off and travel somewhere else and establish sponges elsewhere. They can also be hermaphroditic, so when something is a hermaphrodite, it means that they have both the male and female reproductive organs, and they can produce male or female gametes, depending on, some of them can only produce one at a time, and then many of them can produce both the male and female gametes at the same time. And so they will basically can act as both the males and the females when they're interacting with other sponges. They can reproduce internally or externally. So they can either release their sperm and egg into the water column and hope that it meets up with sperm and egg of the same species, or some sponges will keep the eggs within them and the males will release the sperm, and the sperm will go and meet up with the eggs within the female, and that's internal fertilization. Sponges are also able to regenerate, so you can break off pieces of a sponge, or you can even divide a sponge into pieces and put those pieces close together, and they will stick back together in the correct orientation. The next phylum is Cnidaria, and cnidarians include jellyfish, anemones, and corals, and some other organisms. And cnidarians are protostome invertebrates, so their mouth develops first, and they have no backbone. And they actually have an incomplete gut. So the opening that is their mouth also acts as their anus as well. Unlike the sponges, cnidarians do have true tissues, so they do have aggregates of cells that work together to perform a common function. Cnidarians exhibit radial symmetry, so we can divide them in half using multiple bisecting lines and get two equal mirrored halves. And then the cnidarian life cycle is composed of two body plans. They have the polyp form, which looks something like this. And this is the form that is generally sessile. Some of them can move a little bit, but they're not really doing a lot of moving. Most of them are attached, and they don't leave wherever they're attached. And then they have the second form, which is the medusa form. And the medusas are the motile form. They can swim to various different locations. They're not super strong swimmers, so they're often carried away by currents, but they can swim. And many of you are familiar with these as jellies or jellyfish. The life cycle of many cnidarians includes both of these stages. So at some stage in their life, they're at a polyp, and then some stages they're medusa. But there are some species that only exist as polyps, like sea anemones or corals, and then some that are only exhibited as medusas, like box jellies. Cnidarians are carnivores, so they eat other animals and plankton and things like that in order to get the food they need, mostly small crustaceans and fish. And how they do this is they have tentacles around their mouth that they use to grab food. So in this image here, the mouth is here. It's a bit hard to see, but you can see it in this picture of this anemone right here. That's the mouth. And then the mouth is surrounded by a variety of tentacles. And you can see those here, right? And same thing here on this anemone. All of these kind of whitish, pinkish projections are the tentacles. And on their tentacles, they often have stinging cells that they can use to basically shock their prey. And then they'll use the tentacles to bring the prey into their mouth and into their gut. And this is the gut. This cavity here is their gut, and then they basically digest the food inside the gut, and then any of the waste that's left over, they push it back out of the mouth. So I mentioned they have stinging cells. These stinging cells are called cnidocytes. Sorry, let's erase some of this. Okay. Those stinging cells are called cnidocytes. And an example that many people are familiar with, if you've ever been to the beach and got stung by a jellyfish like I did recently, are nematocysts. And so these stinging cells are unique to cnidarians. No other organism has these naturally or makes these themselves. Some organisms that eat cnidarians will recycle their cnidocytes and use them for their own purposes, but they don't make them. Only cnidarians do. So keep that in mind. So how do these cnidocytes get triggered and kill or debilitate their prey? Well, how this works is the cnidocyte is composed, it's a single cell that's composed of a trigger, which you can see here, and I'll erase it so you can kind of see it. It's kind of small. There we go. So they have a trigger, and then they have a very, very tightly wound spindle and a barb. And it's a little bit challenging to see, but in these next image you can see it. So when the trigger on the cell is touched, let's say by an unsuspecting person at the beach, like myself or many of you, that will allow the cell to open up. And basically the water will rush into that cell, and it'll cause the barb and the barb to spring out because the string is under, that fiber is under such high pressure. The barb will rush out, and so that's what that's showing you here. And it will inject itself into the prey. So this blue is actually the prey's skin. And so once the barb is in the prey, it will sometimes inject toxins, and that will cause the animal to go limp or to straight up die. And so that's how these jellies and anemones and corals can work to catch their prey. I mentioned that the phylum Cnidaria includes corals, anemones, and jellies, and so we're going to spend a little bit of time looking at each of those. So corals are large colonies of very, very small polyps, and I included a picture here of a coral polyp. So they're just a whole bunch of very, very small polyps that are really, really tightly packed together, and they're all attached to a hard exoskeleton. So there's a skeleton underneath that's made of calcium carbonate that they all stick to. Corals are sessile, so they're formed and they don't move, and that is partly because of their calcium carbonate skeleton is not living. It's just a hardened mineralized structure that is attached to a substrate. They can reproduce sexually or asexually, and they can undergo internal or external fertilization depending on the species and the conditions. And then something that's really important to note about corals is this phenomenon called coral bleaching. And coral bleaching is when essentially a coral will lose all of its color and it will die. And this is being attributed to climate change. So corals have the polyps have a close mutualistic relationship with these small photosynthetic protists called zooxanthellae. And you can see them in this picture here, as all of this orange material is actually microscopic protists that live within the polyp. And those protists are responsible for undergoing photosynthesis. And the polyp benefits because when the organisms undergo photosynthesis, the carbohydrates that they produce, the polyp then can consume and use to, survive. And then the zooxanthellae benefit because they get protection from other organisms that want to eat them, and they also get a nice, safe place to undergo photosynthesis, right? And so what happens is, as the climate is changing and temperatures get warmer and warmer, this causes the ocean temperatures to increase as well. Well, these zooxanthellae do not like really warm temperatures. Some can handle warmer temperatures than others, but if the temperature gets too high, they basically start to produce toxic compounds. And those toxic compounds will make the coral sick. So the polyp, in an effort to save itself from these toxic compounds, will basically get rid of its zooxanthellae. And so what happens is, as the coral gets rid of all of its zooxanthellae, it'll go from being something like this, which is a nice, vibrant color, to looking like this, and it'll be white. These polyps really, really benefit and actually need the zooxanthellae to survive because they help to supplement their diet. Without the zooxanthellae, the coral will die. And in some cases, if the water temperatures decrease for a long enough period of time, the zooxanthellae may return if the coral polyps are still alive. But if the coral polyps have since died, or if the temperatures don't decrease far enough, then those corals stay bleached forever and they just straight die. And so this phenomenon is getting more and more prevalent, especially we can see it in like the Great Barrier Reefs and some of the reefs that are in the Atlantic and all over the world really. that they're becoming more and more bleached. And this is problematic because a high percentage of marine and freshwater fish spend some time of their lifetime in corals. Some of them live in corals and they never leave, but a lot of fish will go lay their eggs and raise their young in corals and then travel elsewhere. And so if corals aren't around, then there aren't any fish reproducing, and there aren't any fish for larger organisms like sharks, whales, and especially humans. So a lot of the fish that we enjoy, that we eat all the time, come from they spend some part of their life cycle in the corals. Some other types of cnidarians are sea anemones, and the sea anemones always exist as a polyp. They're similar to what we see in corals, but they are much larger, and they don't behave the same way, right? They don't have a exoskeleton on the bottom or anything like that. And some of them are mildly motile, so they can kind of come off of the substrate and move around a little bit. They're not gonna move around very fast, but they do this to try to get away from predators whenever they can. And you can see a picture of a sea anemone. A picture of a sea anemone here. A good example, if you've ever seen Finding Nemo, the fish marlin and dory and all of them end up living in a sea anemone. And then another example are the jellies. And our jellies can be seen here. This is a box jelly, and they're the medusa form of the organism, and some of them can even be lethal. The box jellies have a pretty powerful sting. Portuguese man-o'-wars are also pretty nasty. Vast majority of stings by cnidarians won't hurt you. Some of them, like if you touch a lot of sea anemones, you might not even feel it. But for some of these jellies out there, it can really be problematic if you get stung. The next type of organisms we're going to talk about I've entitled worms, worms, and worms. And this is because instead of talking about one organism by itself, we're going to talk about 3 organisms kind of together. And this includes the flatworms, segmented worms, and roundworms. So the term worm is actually a very general term for pretty much any small, slender organism that can move around. But the term worm is used to describe 3 very different types of organisms. And so the similarities that we see in these 3 very different types of organisms is actually due to convergent evolution. So these 3 types of worms do not share a recent common ancestor. They actually have developed similar body plans due to selective pressure. There are 8 phyla of worms out there. 3 that we're going to talk about are the annelids, nematodes, and platyhelminthes. All worms, though, are protostomes, so they develop mouth first. They are invertebrates, so they have no vertebrae, no spine, no backbone. And they do have true tissues, and they have organs. So when we talked about sponges, they had no tissues, no organs. Cnidarians had tissues, and then now these worms all have tissues and organs. All the animals we'll talk about from this point on also all have tissues and organs. And then additionally, all the worms are bilaterally symmetrical. So they all have one line that you can basically divide them by and get 2 equal mirrored halves. The first phyla of worms we're going to talk about are platyhelminthes. And platyhelminthes are often referred to as flatworms. And they're referred to as this because they are flat. They're really flat. These platyhelminthes don't have a body cavity that separates their digestive tract from their body wall. All the other worms do. They also do not have respiratory or circulatory organs. And that's simply because they don't need them. They're so flat that all of their cells can just undergo diffusion with their environment to exchange gas and to remove waste and things like that. So they don't need a circulatory or a respiratory system. In order to grow, they add to their body mass, so they do not molt, and many of them have an eye spot to help with navigation. And I'm going to try to draw a picture of what a planarian looks like, because I didn't include a picture here, so you can kind of know what I'm talking about. But I do recommend Googling it, because we've seen my drawings, they're not that great. So a planarian looks something like this. Okay, the worm is something like that. They have a head region, and on their head region, they have two dark, like, oh gosh, that's really hard to see. They have two dark spots, and those dark spots that you'll see in those pictures are the eye spots. And they can use these for navigation because they can sense where light is coming from. Most Platiomethes are hermaphrodites. Some aren't, but most are. An example that you can see that they're not hermaphrodites is in this image here. This is a blood fluke, and the outer worm here that's kind of more spiky looking is the male, and then this one that's in the center is the female. And they stick together basically for their entire lives and mate that way. So the female lives within the male. But for the vast majority of worms, of Platyhelminthes, they're hermaphrodites. And then Platyhelminthes can undergo both sexual and asexual reproduction depending on the species and the conditions. Platyhelminthes get a lot of attention from humans because a decent bit of them are parasites for humans. So an example can be seen with this blood fluke, like I mentioned, that's down here. Another example can be seen up here. This is a tapeworm, which is also a human parasite, but not all of them are parasitic. Some are free-living, and the free-living forms can be seen kind of like this image here, are generally really, really beautiful to look at. They live in marine environments, and they're carnivores, so they're active hunters. The ones that are parasites, though, often have two hosts. So, like for example, with a tapeworm, their first host may be some sort of farm animal like pig or cow or something like that, and their second host is us. Same thing with liver flukes, blood flukes, various different parasites out there that belong to this group. And then all of these Platyhelminthes have a couple of different types of guts. They can have a blind gut, so one way in, one way out. They can have a complete gut, which where food goes in the mouth, leaves through the anus, or they can have no gut. And an example can be seen with the tapeworm. Tapeworms don't have a gut at all. They just absorb the nutrients from your digestive tract that you worked so hard to break down. They just basically steal it from you. Another group of worms are those that belong to the phylum Analeta, and this includes what we call our annelids or segmented worms. And they're called this because they have a segmented body plan. And you can see it pretty good when you look at this picture of this earthworm here. If you look at this part of the body, you can really see these lines in their body, right? Each of these lines. And all of those lines are segments of their body. And in order for these types of worms to grow, they just add new segments to their body, so they do not undergo molting. There are three groups of segmented worms. We have our polychaetes, and they're called this because worms can have these like small hair-like bristles on the outsides of their bodies. Earthworms don't have them, or they do, but you can't really see them. They're very few. But some of them have a whole bunch of them, and essentially what they'll look like is say you have a worm, and they'll be covered with these little hairs. Okay, and those little hairs are called setae. And so if a worm has a lot of those setae, or a lot of those little hairs, then they're called polychaetes, which means many setae. And most of these are marine worms. And some examples are like fan worms and fireworms. And a fan worm, if you've never seen one, is this organism here. And it looks like a giant feather, or it looks like some sort of plant underwater, but it's actually not. It's a worm. This part that you see here are its feeding structures that it uses to catch food. The actual body of the worm lives down here in this tube. So if you were to pull at the tops of that organism, if you could catch it, they're really fast, you would see a worm come out of that tube. Then we have another group are the oligochaetes, and oligochaetes just means few setae. So they also have these bristles, but they don't have as many. An example are earthworms. For the most part, they look bald, but if you look really, really, really close, they have a few of these little bristle-like structures on their bodies. And most of these are terrestrial or live in freshwater environments. And as I mentioned, an example are earthworms, which if you've ever grown any type of garden or any plants outside, or if you've ever gone fishing or done agriculture, you know how important earthworms are, and you've seen these guys. Or been outside after it rains. That too, you also see them. And then we have our last group are leeches. And many of you are familiar with leeches. If you're not, go ahead and Google them. But these are can be predators or parasites. So some leeches are active predators that go out and they hunt other organisms and consume them. And then some of them are parasites where they'll attach to a host and drink the host blood until they're full and then they leave. And so, like I mentioned, some examples of these types of worms that fit within this classification, this phyla of Annelida, are fanworms, earthworms, and leeches. Then the last group of worms we have are our nematodes. This is phyla Nematoda, and these are our round worms. And they're called that because they're very, very round. And it's because they have a lot of pressure that's built up in their bodies. Nematodes are ubiquitous. They are everywhere, and they're incredibly abundant. They're from freshwater environments to terrestrial environments, on the skin, in your body. They're even in the Antarctic, and they're in the lowest parts of the ocean. These guys are everywhere. So they're very, very hardy, and they're very, very ubiquitous. They do grow via molting. So this is the only group of worms that need to molt in order to grow. And they can be free living or parasitic. So there are some free living versions that will basically just hunt and eat different bacteria in order to survive. And there's parasitic versions that can parasitize both plants and animals. And then when they're parasitizing animals, they're of importance a lot to like doctors and epidemiologists and things like that, because they can cause some serious illnesses in humans and in our pets. So heartworm, if you've ever had a pet and they tell you make sure you go and get them treated for heartworms, this is really, really important. This is a type of nematode that basically will live in your animal's heart. And if you ever look up a picture, it's really, really sad what happens to their hearts. So always get your animals treated. Heartworms can also infect humans too, so keep that in mind. The animal that causes something called elephantiasis in people, or trichinella as well in people, are also caused by nematodes. The next big group, which is the second largest group of animals that exists, are the molluscs, or those that belong to the phylum Mollusca. And this includes clams, octopi, squids, and snails, and various other organisms. Like I mentioned, this is the second most diverse group of animals. This is only second to arthropods. Arthropods are the most diverse. Mollusks are protostomes, so their mouth develops first before their anus. They are invertebrates, so they have no backbone, and they do have true tissues and organs. And they also exhibit bilateral symmetry. And when they're growing, they grow by adding new segments to their body plan, so they do not undergo molting. Some key characteristics of mollusks that all of them share is some sort of shell. And so the shell in some of these organisms is very apparent, like in snails, but sometimes they're a little bit less apparent. So like in squids have a shell. It's just underneath the outside of their body, so you can't see it. And then some have lost their shell. So octopi are an example of a mollusk that does not have a shell. And they come from a shelled ancestor, so all of their ancestors had shells, but they lost them at some point in their evolution. Additionally, mollusks have a mantle, which is basically a tissue layer that encompasses all of the organs, and it also is responsible for secreting the shell. And the mantle in like this image here of this this snail here, the mantle is hard to see, but it's this thin layer right here that lies right below the shell. That's the mantle. And it makes the shell and it protects the organs. In something like a squid, the outside portion that you see of the squid, the body of the squid, like the part that makes up what you would think of as the head of the squid, is the mantle. So sometimes the mantle can be on the outside of the shell, and sometimes the mantle is underneath the shell, depending on the different type of organism. And then they all have something called a radula. And a radula is a tongue-like structure that is covered in little bitty teeth, and they basically use this to catch food. So like in the case of snails, they use their radula to scrape off algae from rocks and other hard surfaces. In the case of a squid, they use their radula to break down their prey and then to remove the flesh from their prey. Mollusks exhibit a lot of diversity. They can be marine, like we see in squids and octopi. They can be freshwater, and then they can also live in terrestrial environments like snails and slugs. And some examples are gastropods, which are your snails, and then you have your bivalves, and then you have your cephalopods, which we'll talk about each of those in the next couple slides. The term gastropod means belly foot, and they're called this because they have an enlarged foot that they use for locomotion. You can see that as this part of the snail, and their feet are almost indistinguishable from the head, but that's kind of the foot, and then same here. This is kind of their enlarged foot that they use to get around. Many of them have a shell, and that shell is actually one piece, and their shell will curl. So you've probably seen a shell, a snail shell, and it has this is the opening, and let's say this is the shell, and it'll look like this, that kind of curl look. Some of them have lost their shell, so slugs are an example. And nudibranchs, they don't have a shell. And then some of them, in order to protect themselves because they have lost their shell, will use recycled toxins from other organisms. So I mentioned that cnidocytes are only made by cnidarians. Well, some of these marine slugs, like this guy here, called nudibranchs, will eat cnidarians. And so when they eat them, they can repurpose their cnidocytes and use them for their own protection. And some of them also make various toxins and will discourage an organism from trying to eat them in the future or eat any of their kind. And then they can be also terrestrial or aquatic. As I mentioned, there's some that are marine. We all know of aquatic snails as well as terrestrial snails and aquatic slugs and terrestrial slugs. Bivalves are the group of mollusks that contain two shells, and the shells are connected by a hinge. So if you've ever cooked clams, scallops, oysters, or mussels, you've definitely seen that hinge that connects the upper and lower shell of their body, kind of like a fleshy material at the back of the shell. And mollusks can live in either marine or freshwater environments, and they're all filter feeders. And so how they feed is they have a siphon. So I'm going to try to draw this guy. So we have our top shell and our bottom shell. And then they have two siphons that come out of the organism. So we have a siphon that comes out, and another siphon. And so basically these tube-like structures extend out into the water column. One will be responsible for bringing water in to the animal, and the other one will be responsible for pushing water out. So this one siphon brings water in, and when the water is coming in, there's lots of organic material that's in the water. And then inside of these mollusks, they have specialized gills. And these specialized gills are helping them to breathe, but they're also for trapping food. So when that small particles from the water hit the gills, they get stuck. And then the organism basically will use mucus to move the stuck food in the gills to their mouth, and then they eat it. Any water that they don't need that's already been filtered then leaves through the excurrent siphon. So just as a reminder, water goes in when it carries a whole bunch of organic material. The organic material gets stuck to the gills and is later eaten, and then all the water that they don't need is released through their excurrent siphon. And then one way that you may be familiar with some bivalves is through the production of pearls. So we mentioned before that there's a mantle in mollusks that's responsible for producing the shell. In the case of a pearl, we'll say here's our mantle and here's our shell. There is a little bit of a space between the mantle and the shell. If a piece of sand gets in between that space, as the organism keeps growing over time, it'll basically cover that piece of sand with kind of like this mucousy, hard shell-like material. And over time, that builds up over that grain of sand into a pearl. So that's how we get pearls. It's basically an irritant for the bivalve that they're trying to get rid of. The last group of mollusks we're going to talk about are the cephalopods. And the name cephalopod means head-footed. And they're called this because their feet are directly attached to their head. So their tentacles are directly attached to their head portion where their feeding structures and their eyes and other things are located. Most cephalopods have lost or reduced their shell. So you can see that in octopi have completely lost their shell. And then in squids that have a highly reduced shell. So this is an example of a squid. And instead of having a big coiled shell, they have a very thin layer of shell that is underneath their mantle. So you can't see it unless you cut the squid open and open them up, and then you'd see it. And then that reduced shell in squids is actually called a pen. And then some of them still have maintained a large shell that is ancestral to this group. And an example of that are the nautiloids. So this chambered nautilus here has a big, defined, pronounced shell, which is all of this region here. And so they've retained this structure. However, most cephalopods have either reduced or completely lost their shells. Cephalopods are active hunters. They're all carnivores, and they use their tentacles for hunting. So you can see these tentacles here in this octopus. You can also see the tentacles in this squid, and in the chambered nautilus, all these tentacles here. And they can use these tentacles for catching their prey and holding them, and then bringing them closer to their mouths. They can also use their tentacles for locomotion, so they can use them to basically walk or swim to get from place to place, or to help them catch prey or escape predators. Some of them have tentacles that have suction cups on them, so you can see that in the octopus here. All these little circles here are the suctions. And then also in squids, a lot of squids have suction cups on their tentacles, but in like things like giant squids, their suctions will actually have little teeth to help them catch prey. So their suctions on their tentacles will look something like this. There's the tentacle, and then they'll have these little teeth in there that basically grip into their prey's skin so they can't get free. Cephalopods can also use another mode of locomotion besides using their tentacles to walk or swim. They can also use jet propulsion. So they have these siphons, which are similar to the siphons like we talked about in bivalves, but generally a lot bigger, and they're located near the head. So you can see one here in this squid. It's on their bottom side below where their eye is located. And that opening, they can take a lot of water into that opening and push it out very fast. And when you try to force a large amount of water through a small opening, it produces pressure. And that pressure of the water being forced out will propel the organism backwards. And so that whole process of taking water and expelling it out of the siphon at a fast rate through a small opening to push themselves backwards or to a different location is jet propulsion. It's often argued that cephalopods are or are not intelligent. So your book argues that cephalopods should not be considered to be intelligent because the activities that they're doing or the things that we've seen them be able to do are learned behaviors that they've gained from just trying to survive in the wild. So for example, their ability to open jars to get fish that are contained within is not a marker of intelligence in their perspective. Instead, it's just learned behavior from them having to reach into tight caverns or open up different openings to get prey. I would like to argue that cephalopods are intelligent because there's been evidence that they can learn. So there have been cases where a octopus in one tank will watch another octopus in a different tank solve a problem. And then when you give that first octopus the same puzzle, they solve it faster than the first octopus just because they learn from watching the other octopus solve the problem. So it can be argued that cephalopods are or are not intelligent. It's up to the person that you're talking to. Some examples we talked about: squid, octopi, chambered nautiluses, and also cuttlefish, if you know what those guys are. So before I mentioned that the organisms that were in the phylum Mollusca, or the molluscs, were the second most diverse group of animals on the planet. And they were only second to arthropods. So now we're going to talk about phylum Arthropoda. Some organisms that fit within this phylum are insects and crustaceans, but there are various other members of this phylum as well. In addition to being the most diverse group, they're heavily abundant. So if you were to go outside, you would definitely, I guarantee you, if you didn't see an arthropod immediately, you'd see one within a couple of minutes. And this is because they are absolutely everywhere. There are also probably a couple living in your home currently, whether you can see them or not. They're hanging around there, probably like a spider here or an ant there kind of thing. Arthropods are all protostomes, so they develop mouth first. Additionally, they're invertebrates and they exhibit bilateral symmetry. So you can only divide them into two equal mirrored halves using one bisecting line. Arthropods also all have a exoskeleton made of chitin. And chitin, if you remember this term, we talked about chitin when we talked about fungi, and it was the carbohydrate that was in the fungi cell wall. That same carbohydrate makes up the exoskeleton of arthropods. And this chitin exoskeleton is really good for protecting the organism from any types of dangers. So protecting it from predators, also from protecting them from when they fall from great heights, they don't die because that exoskeleton helps to protect them. Additionally, that exoskeleton helps them to conserve water. If they didn't have it, they may dry out because they're terrestrial, at least the terrestrial ones would, because they're exposed to the air, and naturally water wants to move out via diffusion if you're in the air. The aquatic arthropods don't have to really worry about this drying out so much, but they still do have a chitin exoskeleton. And in order for arthropods to grow, they have to molt. So they have to shed that exoskeleton and then grow and then make a new exoskeleton once they've grown bigger. Arthropods are also segmented. So some of them are more segmented than others. In like millipedes and centipedes, like we'll see in the next slide, they have a lot of different segments. But in even ants and things like that have various segments. They have a head, an abdomen, and a thorax, right? So those are segments. In addition to having various segments, they also have jointed appendages, and you can see that really well in this picture here of this organism. So this bug has a couple of jointed appendages that you can see. You can see the joint here on its leg. You can also kind of sort of see the joint here on the bottom part of its leg, and on this side it's easy to see the joints. They also have joints in their antennae. You can't really see it very well in the shrimp, but you can kind of see the joint here on their mouth parts, and they also have joints on their antennae that help them to move around. There are four major groups of arthropods, and this includes the myriapods, which are the millipedes and centipedes, the salicerates or chelicerates, and this includes spiders, scorpions, horseshoe crabs, ticks, and mites. Then you have your crustaceans, which include lobsters, shrimp, crabs, barnacles, and krill. And then the insects, which is by far the largest group of arthropods and includes ants, beetles, dragonflies, moths, butterflies. The list keeps going on and on. Myriapods are long, slender, segmented-bodied arthropods, and they have many feet. You can see them in various pictures. You can definitely see them here. You can see them in this millipede here, as all these little projections under its body are all its little feet. And they can be herbivores or carnivores depending on the type. So when we're talking about millipedes, like this guy, they are herbivores. So they generally live in the forest floor. They eat leaves that have fallen from trees and they're decaying on different plant material. Centipedes, on the other hand, like this guy here, are carnivores, and they generally have fangs, venom, and jaws to help them hunt. So I personally think millipedes are super cute. I love millipedes. Centipedes, on the other hand, no go. Chelicerates or chelicerates, depending on how you pronounce it, include spiders, horseshoe crabs, scorpions, mites, and ticks. And they all have 4 pairs of legs. So they have 8 legs in total, and they have specialized feeding structures. And so if you've ever really looked at a spider, like really, really close up, you'll see around its mouth parts that they have various appendages that help them with breaking down their food. So they have a couple of appendages that help them to maybe inject venom, and they have a couple of other appendages that are responsible for helping to break their food down into smaller pieces for consumption or to hold on to prey, and various other things involved in predation. Additionally, ticks and mites also have specialized feeding structures, but these feeding structures are specialized to penetrate the skin of their potential host and get into the bloodstream so they can adequately suck the blood from potential hosts. Chelicerates or chelicerates can be aquatic or terrestrial. If you go outside, even right now, you can probably see a terrestrial chelicerate. This is like spiders, scorpions, ticks, or mites. Hopefully you don't see any ticks or mites because those things suck, like quite literally. But you can also see some aquatic forms if you go to certain beaches in the form of horseshoe crabs. So sometimes they'll come out onto the land, or if you go scuba diving in some areas, you might see these horseshoe crabs. All chelicerates are either carnivores or parasites. So we talked about the parasites would be mites and ticks, but then the vast majority of them are carnivores and they're active predators. And the carnivorous forms have jaws, venom, and various enzymes to help them capture and digest their prey. Crustaceans are a group of arthropods that you may be really familiar with if you like seafood. This group contains shrimp, crabs, and lobsters, as well as barnacles and krill. And all crustaceans have 5 pairs of appendages on their head, 3 that are used for feeding and 2 antennae that are used for sensing their environment. Arthropods are carnivores, and they can be sessile or motile. So some sessile forms are like barnacles, where they attach to a substrate and then they basically live inside of their home and filter feed from there. Or they can be motile, like shrimp and crabs and things like that. The vast majority of crustaceans are aquatic, so they can live in either marine or freshwater environments, but there is an exception. So the roly-poly bug, or some people call them pill bugs, some people call them wood lice, but they're this guy here, and that crustacean is actually a terrestrial crustacean that feeds off of the dead plant material that is on the forest floor. So they're actually terrestrial herbivores rather than aquatic carnivores. And then the last group of arthropods we're going to talk about is by far the largest and most abundant group, which are the insects. And insects all have 3 pairs of legs, so they have 6 legs in total, and a lot of them have either 1 or 2 pairs of wings. Wings are not required for an insect to be considered an insect, but a lot of them do have wings. And even in some cases, like some species of ants, majority of the members of the colony will not have wings, but a couple of the members of the colony of the same species may develop wings, like the royal ants and things like that. So wings are common. Insects must undergo metamorphosis to be converted from their larval stage into their adult stage. And then the most of the insects are terrestrial, but some are aquatic. And some examples, and these are literally just a few examples, there are so many insects out there, are ants, beetles, bees, flies, moths, dragonflies, butterflies. The list goes on and on and on. Metamorphosis is the process of restructuring an insect larva into its adult form, and it occurs in three major stages. The first stage is the larval form, and so this is usually presented as a caterpillar or a grub. And the responsibility of the larval form is to eat and grow. So they just eat a whole bunch and grow bigger. And then once they're gotten big enough and they've eaten enough and the conditions are right, they will convert to the next stage, which is the pupa form. So the larva will form a hard outer casing on its outside, and that's where it will basically stay until it's converted into an adult. This casing can be a cocoon or a chrysalis, as just as some examples. And within that casing is where the larva characteristics will be broken down and recycled into making the characteristics of the adult. And then finally, once the adult is matured and ready to go inside of the chrysalis or cocoon, or whatever casing that they're in, the adult will leave the casing and go on to live its life. Now, the adult form is responsible for reproducing. So much so that some species of insects, adults, don't even have mouth parts for feeding at all. They literally reproduce and die. Some of them do consume food, so we see here in butterflies, they a lot of times drink nectar, but some species don't even have mouth parts. That's how dedicated the adult form is to reproducing, to make more eggs for more larvae, more pupa, and so on and so forth. The selective pressure that is experienced by caterpillars and butterflies, or grubs and beetles, can be very, very different. And this is because in this process, like we just talked about, is called complete metamorphosis. In this process, the larval form and the adult form are so vastly different that the experiences that they have are very different due to selective pressure. So for example, let's say in a field where this butterfly drinks the nectar from a specific flower. The caterpillar is herbivorous. It's going to eat the leaves of whatever trees it's supposed to, and it can survive. So it experiences certain selective pressures associated with its life cycle. But once that caterpillar develops into a butterfly, if all of the flowers that it normally drinks nectar from are gone, now it's experiencing a selective pressure that the caterpillar did not experience, just due to the simple fact that it has different lifestyles. So just keep that in mind, that these differences between the larva and the adult stage can also result in natural selection having different impacts on the larva and adult stage. So what we were talking about, like I mentioned, is complete metamorphosis. There's also incomplete metamorphosis, and this is where the young form of the insect is not super different from the adult form. They're usually a smaller, more immature version of the adult. They generally will eat similar prey. So if they're adults, eat particularly leaves from a tree, the young generally will eat the same leaves from the tree or insects, whatever have you. The young form of the insect, when they undergo incomplete metamorphosis, are called nymphs. And you can see that here with this grasshopper. This is a grasshopper nymph, and this is the adult. Sorry, poor writing, but that's the adult. Nymph. And you can see even by just looking at them that they're not too terribly different. Now the nymph are immature, so they cannot reproduce, and oftentimes they may lack certain characteristics like wings and things like that, but they don't differ nearly as much as the differences we see when an organism undergoes complete metamorphosis. Some examples of organisms that undergo incomplete metamorphosis are grasshoppers, dragonflies, and roaches. Organisms within the phylum Aquinodermata are the first deuterostomes we're going to be talking about. And so just as a reminder, that means that first opening in their early development becomes their anus. So they develop anus first or mouth second. And some examples of echinoderms are starfish, sand dollars, and sea urchins. They're all invertebrates and they all exhibit radial symmetry. in their adult form at least. Their larvae are actually bilaterally symmetrical, and this indicates that they do have a bilateral ancestor. They are generally covered in spines. You can see that most apparently in sea urchins. So you can see all these little purple puffs, and then this pink puff here are the sea urchins. You can clearly see their defined spines, but other echinoderms such as starfish also have spines. They're just generally a little bit smaller and maybe a little bit more flattened out. They all have a nervous system but no brain, and they have these things called tube feet for locomotion. So you can see an example of tube feet in this image here. All of these white or kind of like transparent projections here are all tube feet, and they can use these to move around. Each of those tube feet can move independently, but you can coordinate their movement so they can project the organism in whatever direction it needs to go. The tube feet also help with predation, so it helps them to catch their prey and hold on to their prey, as well as open up their prey. So if it's like a bivalve or a snail, they can open up that prey so they can put their stomach in there and digest the organism to eat it. Like I was just mentioning, these echinoderms are vastly carnivores. So starfish will hunt a lot of bivalves and snails and slugs and things like that. Sea urchins, they eat kelp, which as a reminder is not a plant, it's a protist. And they can actually decimate kelp forests, which is a problem, especially over in the west coast of the United States, where overgrowth of sea urchins due to a loss of sea otters is decimating these kelp forests and causing major problems in those ecosystems. They can also be filter feeders, as in the case of sand dollars. The largest group of deuterostomes belong to the group Chordata, and Chordata or chordates include mammals, fish, reptiles, and amphibians. The members of Chordata or chordates have true tissues and organs, exhibit bilateral symmetry, and we all share a fish-like common ancestor that existed about 500 million years ago. There are some key developmental similarities for an organism to be considered a chordate. They have to have a notochord, they have to have a dorsal hollow nerve cord, pharyngeal gills, and a post-anal tail at some point in their development. So they may not have to have these features as an adult, but they have to have them in their early developmental stages. There are three groups of chordates. There are the vertebrates, so those that have a backbone, and then there are two groups of invertebrates, which are our tunicates and our lancelets. So as I mentioned before, all chordates share four main characteristics. The first being that they all have a notochord. And a notochord is just a rod of tissue that runs from the head to the tail along the dorsal side of the organism. So you can see it in this picture here as the blue highlighted region. And the notochord stiffens when the organism's muscles are contracted, and it provides support during movement. The notochord later hardens and stiffens in vertebrates to become the backbone, aka the spine. All chordates also have a dorsal hollow nerve cord, and this is just a cord of nervous tissue that runs from the head to the tail, and it can be seen here in this as this blue area, all that blue. The dorsal hollow nerve cord in vertebrates later becomes the central nervous system, including the brain and the spinal cord. All chordates also have pharyngeal gill slits at some point in their development, and these gill slits are basically just folds of skin that occur between the back of the mouth and the top of the throat, and these are indicative of a shared aquatic common ancestor. So these slits were used by the common ancestor for breathing and feeding. However, many chordates, especially those in terrestrial environments, have since lost these gill slits. Some have maintained them and they later develop into gills if you're an aquatic species, but terrestrial chordates don't necessarily need gills, and so in the adult forms, these gills have been lost. Finally, all chordates have a postanal tail. So the extension that goes beyond the anus is the postanal tail. Has disappeared in many vertebrate adults, but all of the fetuses of chordates have a postanal tail, and you can see that here as this blue region. The simplest chordates are both invertebrates, and this includes the tunicates and the lancelets. Tunicates are small marine invertebrates, and they look something like this. So these little blue circular organisms, I think they're actually quite cute. Those are the tunicates, and they're sessile filter feeders. So they usually form on rocks, anchors, sides of boats, docks, things like that. And once they attach, they spend their whole life in that location, and they just filter feed from there. But despite their odd appearance, they actually are chordates, and we know this because of their larvae. So the chordate larvae have all the four major characteristics that are required of chordates. So they have a notochord, a dorsal hollow nerve cord, a postanal tail, and a pharyngeal gill slits when they're larvae. But once they grow as adults, they lose all of those characteristics except for the gills, which they maintain for filter feeding. And then the other group are the lancelets. And the lancelets are slender eel-like invertebrates, and they're also filter feeders. And so you can see a picture of one of them here. And both their larval and adult forms maintain all of the key characteristics of being a chordate. So both the larva and adult forms have a notochord, a dorsal hollow nerve cord, tail, and gill slits. The largest group of chordates are the vertebrates, and vertebrates are characterized by the presence of a backbone that's made of either hollow bone or hollow cartilage, and it surrounds the notochord. The responsibility of the backbone is to protect the dorsal hollow nerve cord, aka the central nervous system. And all vertebrates have a head that is composed of a skull, brain, and sensory organs, and they all have all four required characteristics to be a chordate at some point in their life cycle. So they may have lost some of the key characteristics to be a chordate as adults, but in their fetal stage, they do have these characteristics. The simplest group of chordates are the jawless finless fish, and the common shared ancestor for all chordates is thought to resemble these types of fish. These fish have no jaws and no fins, hence jawless finless, and they swim in like an S-shaped form. So almost like you imagine a snake crawling on land, that's how they swim but underwater. The two types of jawless finless fish that we have are the lampreys, which are seen here, and the hagfish, which are seen here in this lower image. Lampreys, for the most part, as adults, they do not feed. They just live off of stores of food that they acquired when they were eating as youth. But some of them can be parasitic. So people will often find when they go out fishing, they catch fish or sharks and other organisms out there, and you'll find these lampreys attached. And they're just basically feeding off of that fish in order to survive. And then we also have our hagfish, and hagfish are basically decomposers. They eat any dead organism that has died and sunk to the sea floor. They go ahead and start the process of breaking that down. And there's actually some cool videos of these hagfish attacking the remains of a whale that sunk to the bottom of the ocean. So if you're interested in that, I would say go ahead and check it out. It's pretty cool. The next level of complexity are the jawed finned fish. And these fish have jaws for catching prey and consuming prey, and then fins for locomotion. And these fins they're able to use in very specialized ways. So they'll have multiple pairs of fins that they use for different things. They have a pair for propulsion, so for moving themselves forward through the water column. They'll have a pair for equilibrium, for steering, and for stopping. So there are multiple sets of fins, generally in pairs along the jawed fin fish body. They also swim in an S shape like we saw in the jawless finless fish, but they have a lot more control due to all of these fins. They can have more specialized, more controlled movements. There are three groups of jawed finned fish. We have our cartilaginous fish, and this includes our sharks and rays. And the big characteristic of them is that their skeleton is completely made of cartilage. And so the cartilage is what also makes up this part of our nose and what makes up the kind of more structuring part of our ears. So cartilage is sturdy, it's solid, but it's also flexible. So if you pinch your ear and you move it around, you can feel it's kind of hard, but it still can move. And that's one benefit of cartilage, is it provides structure, but it also provides flexibility. And then we have our ray-finned fish. And this includes vast majority of what you would consider to be a fish. When you think of a fish, it's probably this. It includes salmon, goldfish, and trouts. And this is the most diverse group of vertebrates out there. And they're called ray-finned fish because they have fins that are made of bone. Not just their fins, their whole body's made of bone, but they're called Rayfin fish because they have these bony projections that are, you can see here in this picture, all this like green part of this image is showing you the bones. And the bone projections are webbed together by a membrane of skin, which you can see in this part of the image, all the kind of orange skin that's between the bones. And that's why they're called Rayfin fish. There's a ray of bones connected by a membrane of skin. And as I mentioned, they have a body made of bone, not cartilage, and bone is far less flexible than cartilage. So there is a lot more structure there, but a lot less flexibility in these fish. These fish also have a swim bladder, and a swim bladder is basically like a pocket of air that they have within their bodies that has its own trachea and alveoli that allow them to basically be buoyant in the water. So when a cartilaginous fish is in the water column, it must continue to swim to remain, to keep from sinking, basically. And these rayfin fish, they have their swim bladder, which allows them to remain buoyant even when they stop swimming. So when they stop swimming, they don't sink, unlike in the cartilaginous fish. And then the final group of jawed fin fish we have are our lobed fin fish. And they're called this because of the difference in their fin structure. So in the ray fin fish, they had those rays of bony projections connected by skin, whereas in the lobe fin fish, they have a lot more meaty, hardy fins. So you can see the differences in the diagram here, but you can also look at the fins on this actual fish. So if you look at these fins and you compare them to the fin of the ray fin fish, they seem a lot more fleshy and meaty. And the lobe fin fish's fins have a central appendage of bone and muscle. And this is one of the characteristics that is believed to have allowed for the early terrestrial ancestor of all terrestrial vertebrates to move from the aquatic environment onto land. The first land colonizers were thought to have been lobed fin fish that lived in shallow coastal waters near to land. And these lobed fin fish that eventually moved from the aquatic lifestyle to the terrestrial one are the shared common ancestor of all terrestrial vertebrates. So in order to go from an aquatic lifestyle to a terrestrial lifestyle, there are a lot of adaptations had to have occurred. One, the lobed-fin fish had to develop lungs. And so many lobed-fin fish nowadays, you can see, will have lungs and they'll have gills. And gills are great when you have an aquatic lifestyle, either full-time or part-time, but when you're a terrestrial organism, gills are not that useful. And so the common ancestor of all terrestrial vertebrates lost its gills and further developed its lungs to get full-size lungs. The early lung is thought to have derived from early air sacs that were similar to the ones that developed into the swim bladder in the rayfin fish, but instead of becoming a swim bladder, it became some sort of lung that was further developed later on to become lungs as we know them today. Additionally, these lobed-fin fish had to develop a backbone, and a backbone is basically just interlocking vertebrae that provide support against gravity. This is really important because if you live in an aquatic environment, you don't have to fight the forces of gravity so much. because there's water below you, water above you. You just swim through the water. But once you're on land, the pull of gravity is a lot stronger and can have a detrimental effect if you don't have a nice, strong support system. And so the development of a backbone allowed for these organisms to be able to pull themselves against gravity, but to also redistribute their weight. So instead of the weight of the organism being supported by the backbone, instead it was redistributed to be supported by the limbs. The vast majority of land colonizers, the vast majority of vertebrates have 4 limbs, so they're tetrapods, and the early land colonizers had to also develop these 4 limbs. So you can see in the Lobed Fin Fish, it has 4 kind of meaty fins. Those fins later developed into the limbs that we know of today. The bones that make up the lobed fins in the lobed fin fish are homologous to the appendages that we see in terrestrial vertebrates, like mammals and birds and reptiles and amphibians. So that lets us know that we have a shared common ancestor, current lobed fin fish and us. So there was definitely a lobed fin fish ancestor. And then finally, lobed fin fish had to develop a way to prevent their eggs from drying out. If you're a fish, you lay your eggs in water, you don't have to worry about your eggs drying out. It's in water. But on land, this can be a very major problem because your eggs are not surrounded in liquid at all times. And so in order to make their eggs desiccation proof, they developed 2 different new structures. One, the presence of a shell. And the outer shell prevents water from evaporating out of the egg. And then 2, they have a watertight membrane that lies just beneath the shell and encases the organism. And this allows for the amniotic fluid to be maintained around the embryo and it doesn't evaporate out. So all of these characteristics were things or adaptations that the earliest lobed fin fish ancestor of all terrestrial vertebrates had to develop in order to colonize land. And some of these adaptations have since been lost in current modern day terrestrial organisms. So we can see that in in snakes and in whales. So snakes and whales don't have 4 limbs, 4 workable limbs, right? In snakes, you don't see any legs. They're not tetrapods. And same thing in whales. They've lost their 2 hind limbs. Those limbs are still, remnants of them are still present. They have been highly, highly reduced. You can still see the rudimentary bone structure in these organisms, but they're no longer useful. So they've lost those legs secondarily. Additionally, we can see some of these losses in amphibians. Amphibians have lost their desiccation-proof eggs. They do not have eggs with a shell or with a membrane, so they have to lay their eggs in water, otherwise they'll dry out. So our shared common ancestor among all terrestrial vertebrates had all of these characteristics, but some terrestrial vertebrates have lost those characteristics over time. Amphibians were the first terrestrial vertebrates, and the term amphibian just means bearer of 2 lives. And they're called this because part of their life cycle, when they're juveniles, breathe water. And then when they're adults, their second part of their life cycle, they breathe air. And like I mentioned in the last slide, amphibians are non-amniotes, so they are terrestrial vertebrates that do not have a desiccation-proof egg. So that means their eggs do not have a shell or a waterproof membrane. As a result, they must lay their eggs in or around water. And so you can see their eggs here. They look far more like all of this here. They look far more like fish eggs than they do like a snake's egg or a turtle egg. And if they don't lay their eggs in water, they must be near to water. So some frogs will lay their eggs on leaves, and then they will constantly get water from a nearby source and douse the eggs in water to keep them moist. Otherwise, if the eggs dry out, the embryos will die. Some amphibians have lost their lungs secondarily. Some have lungs and can use their skin to respire, but for those that have lost their lungs completely, they rely solely on gas exchange through their skin in order to survive. And this is important to the texture of their skin. They have to make sure that their skin is always moist for gas exchange to occur. If they become too dry, they will basically suffocate. Some have lungs and they can respire through their skin, so they can use both of those mechanisms in order to undergo gas exchange. The fact that the skin is so porous and sensitive to the environment is why we consider amphibians to be an indicator species. And essentially what that means is we kind of use the status of amphibians to know if an area is polluted or not. So if you go out into a nice wetland, then you normally expect to see lots of amphibians, and you don't see any, or you see a lot of dead ones, or you see a drastic decrease in the number of amphibians in that area, then that might be an indication that the area is polluted, because they're so sensitive to pollution that changes in, like, even a little bit of pollutants in their environment can drastically change the population dynamics of amphibians in those regions. So amphibians are a really key indicator on the health of particular environments. Some examples are frogs, toads, and salamanders. Unfortunately, a large quantity of amphibians are in danger. About 1/3 of them are classified as endangered or threatened, and this is due to a variety of causes. There's changes in the environment, so there's climate change, there's loss of their habitat due to humans encroaching upon it. There's also changes due to pollution, so if there's any dumping or diversion of chemicals into certain habitats, they can be drastically affected that way. And they're also affected by disease. A lot of fungal pathogens can go through and completely decimate a amphibian population. The next major group of terrestrial vertebrates are the avian and non-avian reptiles. And avian and non-avian just means bird and non-bird. And birds are considered reptiles, so they're grouped together with reptiles because birds are actually descended from dinosaurs. And so their closest extant relative are crocodiles and alligators. And that's because crocodiles and alligators are the closest thing to dinosaurs that we have that lives today, besides birds. So you can see their shared common ancestor is here. So it's relatively recent. Both avian and non-avian reptiles are amniotes, so they're terrestrial vertebrates that produce an amniotic egg. Their egg has a watertight membrane and a shell. The shell can either be hard or soft depending on the organism. Some turtles and some snakes will lay softer, more rubbery eggs, whereas others will lay harder shelled eggs. All reptiles, both birds and non-birds, have similar skulls, similar leg structures, and they share a lot of genetic similarities. Some examples are lizards, birds, turtles, snakes, and dinosaurs. And of course, there are a lot of differences between the birds and the other groups of reptiles. For one, birds have feathers and they can fly, and then they also are endotherms. So they're able to keep their bodies warm or maintain their body temperature via the energy from cellular respiration. So they can maintain that energy to keep their bodies warm, whereas other non-bird reptiles cannot do this. So they're considered to be ectotherms. So in order for them to increase or decrease their body temperature, they must spend time in the sun or out of the sun. So if they're cold, they must go into the sun, and if they get too hot, they must go into the shade. So endotherms, birds, ectotherms, reptiles. The last group of terrestrial vertebrates are the mammals, and all mammals share a common ancestor. This ancestor was small, nocturnal, and ate insects. We also have a couple other similarities. For one, we all have hair, and hair is just composed of dead cells that are filled with keratin, and it's responsible for helping us to insulate ourselves. We also have mammary glands in the female sexes, and that mammary glands are responsible for making calorie-rich milk for the young as they're growing and developing. Additionally, we have limbs that are located beneath our bodies rather than on the side. And if you want to know an example of this, look at any lizard or alligator or anything like that, and you'll notice that their limbs hang out on the side of their bodies. And this is why when they're running, they have like a wobbly motion. But with mammals, ours are right beneath our body. And this is thought to have allowed us to run faster and farther than, let's say, reptiles. But this faster movement and farther running require a higher metabolism. So you have to have higher metabolism so you can get more energy so you can run faster and farther. This high metabolism also results in more energy, more heat being produced. And that heat we're able to keep within ourselves pretty well. So we're also endothermic. So we use the heat from cellular respiration, aka metabolism, to keep our bodies warm. And all of these things combined have allowed us to be as successful as we are. So especially the endothermy has allowed us to colonize places where other organisms may not be able to live, like reptiles and things like that. Most mammals are viviparous. So this means that they give birth to live young. There are two groups of viviparous mammals. This includes the marsupials and the placentals. Marsupials give birth to underdeveloped fetuses that then further develop in a pouch. So basically, the underdeveloped fetus will then leave the reproductive tract, crawl into the pouch, and attach to a nipple, and it will stay and suckle on that nipple for several months until they fully develop. And then once they're fully developed young, they can leave the pouch. And that differs from placentals. So placental mammals have a placenta, which is responsible for facilitating nutrient, gas, and waste exchange between the parent and the fetus, the mother and the fetus. And then also placentals do not give birth to young until they're fully developed, unless something has gone wrong. So for the most part, placentals have a placenta, give birth to fully developed young, whereas marsupials give birth to underdeveloped young that must complete their development in a pouch. And then there is the odd man out, which are the monotremes. And the monotremes lay eggs. They are very unique in the fact that in marsupials and placentals, we have an amnio ancestor. So our ancestor laid eggs, but we do not anymore. But the monotremes have maintained that ancestral characteristic and they still lay eggs. But they also still have mammary glands. But instead of their mammary glands being connected to a nipple like it is in marsupials and placentals, it's instead connected directly to the outside of the skin. So their young must just come up to the skin and lick up the milk that is being secreted from the mammary glands directly from the skin, rather than latching onto a nipple. And some examples of monotremes are platypi or platypuses and echidnas. And if you've never seen an echidna, it's this cute little guy right here. One group of mammals we're going to spend a little bit more time talking about are the primates. And primates, the shared common primate ancestor arose somewhere around 55 million years ago. And this ancestor was arboreal in nature. And we know this because all primates have some shared common characteristics that indicate that our ancestor must have lived in trees. For one, we have forward-facing eyes with binocular vision. And this is really helpful if you live in trees. So you can assess depth perception. If you're living in a tree, you don't want to swing from tree to tree and miss and fall to the ground or hit another tree. Depth perception matters a lot if you're living in trees. Additionally, we have rotatable shoulders and elbow joints. And this is really helpful when you're swinging from tree to tree or living within trees and traveling around in trees. And then finally, we have opposable digits. So we have opposable thumbs and we have fingers and toes. And these appendages are really good for grasping onto branches. So all of these together, the forward-facing eyes, binocular vision, the rotatable joints, and the opposable thumbs and digits are very good indicators that the shared common ancestor of all primates lived in trees. Some examples are humans, lemurs, apes, and chimps. So a common misconception that's out there is that humans descended from apes. That is not true. We are not related. We are not A gorilla will never become a human. A lemur will never become a human. That's not how it works. We are related to other primates, including apes and monkeys, but we are not descended from them. So that means we have a shared common ancestor, but we did not come from them. So our closest common ancestor are chimps, which you can see this guy here. And you can see on this phylogenetic tree that the chimps and humans share a more recent common ancestor to each other than either of them do with any other primate. And this is also strengthened by genetic similarity. So chimps and humans share a large amount of genetic similarity and even genetic identity with one another. So a large percentage of our genes are identical. Our most recent shared common ancestor lived around 6 million years ago. So this guy in the red dot would have been existed about 6 million years ago. So not super recent, but not super long ago either. And then the human lineage of everything has some distinct characteristics that differentiate us from other primates, including chimpanzees. The fact that we are bipedal, we are larger in size than chimps, and then we also have larger brains than chimps. So when the shared common ancestors between humans and chimpanzees branched off, the human lineage underwent some key adaptations that resulted in humans as we know them today. The first event was bipedalism. So humans, unlike chimpanzees, we walk on 2 legs, whereas chimps walk on 4. And this is thought to have helped humans because it is less energy expensive to walk on 2 legs than it is to walk on 4. Additionally, it also had allowed for us to free up our hands. So if we're able to free up our hands while we're walking from location to location, we can be doing other things as well, like using tools, carrying food, carrying our young, and various other things. So this allowed us to be a little bit more flexible in the things that we can do than our closest relative, the chimps. So the first humans that branched off from a chimpanzee human common ancestor and had these characteristics of bipedalism belonged to the group Australopithecines. And these humans were very small. They were chimp-like, and they lived around 3.5 to 4 million years ago. And they were foragers, so they survived mostly on leaves, nuts, and fruits. And they were partially arboreal. So they lived on the ground when they needed to find food and sometimes to get from place to place. But they spent time in the trees to escape predators and for sleeping. And so this image here is a predicted image of what Lucy looked like, and she belonged to this group. So after bipedalism, the next big step in human adaptations was the enlargement of our brains. So the human brain is twice the size of brains in chimps. And there are various species of humans, and they all belong to the genus Homo. And we all have enlarged brains compared to our shared common ancestor with chimps, and larger brains than our closest relative, which are the chimps. And as the different species of humans, they all have different skull shapes and different brain sizes, with increasing brain size as we got closer and closer to like present day. So this genus of humans includes modern humans, which are Homo sapiens, and it also included early members with large brains but still small compared to what we have today. And then they were also able to use tools. So all the members of the Homo group could use tools, even if they had smaller brains than what we have today. And they migrated from Africa to Europe. So there were various Homo species that all originated in Africa, and then over time they migrated to Europe and Asia, and then eventually to the Americas as they became more and more mobile. And then we as humans have larger bodies than chimps. And this is correlated with the increase in brain size. So as we see an increase in brain size, we also have seen an increase in body size. And then more the brain grows, the body grows. So they're correlated together. Homo sapiens are the only species of living Homo genus. So all the other human species have since died off. Homo sapiens, we are the only species left. Homo sapiens diverged from a similar species, a related species called Homo Erectus, approximately 200,000 years ago. And this all occurred in Africa, which is where all of the Homo species or all the hominids originated from. And then approximately 140,000 years later, the small groups of Homo sapiens began to branch off and travel to other places. So they first started traveling up to Europe and then up towards Northeast Asia and then Southeast Asia. And then in another couple thousands of years, around 15,000 years ago, they crossed the Bering Strait, that is the land bridge that connects Northeast Asia to Northwest America, and they crossed the land bridge and colonized the North Americas and then subsequently the South Americas. As they were traveling, they encountered other hominids or other species in the Homo genus. Some of these include Homo Neanderthales, and Homo Neanderthales are what we refer to as the Neanderthals. They were similar in size to Homo sapiens, but they were far more muscular. And they also lived in organized groups. And they likely hunted large mammals with short spears. And this living in groups and hunting large mammals with short spears is indicated by the presence of severe breaks that we see in the remains of Homo neanderthalis, which suggests that they were probably hunting large animals and got trampled and got these severe breaks, and then those breaks healed. And in order for them to heal, that means they had to have a supportive community that was helping them as they went through their recovery. Then some other species include Homo erectus, which were same size as Homo sapiens, but they had a smaller brain. And there's some evidence that indicates that they likely used fire, and they likely also built boats to travel between different islands. And then finally we have Homo floresiensis. And Homo floresiensis were small-bodied, small-brained Homo groups. So Homo floresiensis, Homo erectus, and Homo neanderthalis and Homo sapiens all lived at the same time. So same genus, but different species. Today, as I mentioned, Homo sapiens, we are the only ones left of the Homo genus. And some of those other Homo species live on in us in our genes. So when we look at the DNA of modern day humans, we do see evidence in our genes of other hominid species due to there was mating between Homo sapiens and like Homo neanderthalis over time, and that's how their genetic information ended up in our genetic code. Congratulations, you made it to the end of our lecture on animal diversification. And I know that this lecture was very lengthy and contained a lot of information. So as you're going through and reviewing, make sure that you break this lecture up into manageable size pieces. So I've broken it up into 2 pieces because I can't upload as one, but feel free to take the part 1 and break it up into 3 different digestible pieces, and same thing with part 2, or however many pieces you need in order to study effectively. So now that we've reached the end of the lecture, go ahead and read chapter 13, sections 1 through 9, and then 11 through 20. And then also, if you have the Launchpad account, go ahead and do the chapter 13 learning curve assignment. And then I've included some videos here that I thought were interesting on sponges feeding, how the jellyfish cnidocytes are, how they function and how they get discharged. Some videos on the tube feet of echinoderms, frog metamorphosis, Lobed Fin Fish, and that was actually really interesting because it shows how people have done experiments with modern day Lobed Fin Fish to see if they can be adapted to travel better on land than their aquatic counterparts. And it basically provides more support for why the common ancestor of all terrestrial vertebrates was very likely a lobed fin fish. And then also a video here that does a good job of explaining the different types of births in mammals. So the monotremes versus the marsupials versus the placentals. So when you're going through this lecture, there's a lot of terminology that you should know. Make sure that you know any of the major terms, especially the ones that I mentioned over and over. So vertebrate versus invertebrate, to know deuterostome, protostome, bilateral versus radial symmetry, like all that terminology you should know. So any terminology that I've especially used more than once, be sure to know those things and then go back and learn all the other terminology you might not already know. In addition, make sure that you know the different phyla of organisms and some examples that fit within them, and then some key characteristics that differentiate that phylum from other phyla. So a good example is the phylum Mollusca includes all the mollusks, which include gastropods. An example are snails, and it includes the bivalves, an example being clams, and then the cephalopods, which an example can be an octopus. And then the key differences between each of those groups. And then know that the animals that fall in the group, the phylum Mollusca, have a radula, a mantle, and had a shell at some point. So something like that. Make sure you start by knowing those types of things, and then going in and learning at more depth. So learning all this material is going to be multi-step process. Going through, studying the big picture stuff, then going through, getting a little bit more detailed, going through, getting a little bit more detailed in order to learn it all. All right, with that said, congratulations, you made it to the end of this lecture, and I'll see you next time.